Okay, here we go. Meiosis video number 2. As promised, we need to talk about fertilization. So fertilization is when we have a union of gamete, egg, meat, sperm. Pretty simple. And it's not just egg, meat, sperm, but we have that influx of DNA from the sperm into the egg. Once that happens, we have a fertilized egg, and that is now called a zygote, Z-Y-G-O-T-E. And that zygote will have one set of chromosomes from each parent. Excuse me. So a zygote produces all the somatic cells by mitosis, and that's how we're going to develop into an organism and into an adult. Okay, so at sexual maturity, the ovaries and the testes start producing from a 2N cell, from a 46 chromosome cell, they start producing haploid gametes. Remember, gametes are the only types of human cells that get produced by meiosis. All the other cells are generated through mitosis. And in meiosis, we have one set of chromosomes in that gamete. It looks something like this. So we have a mature individual, we have a multicellular diploid adult. Those cells have 2N or 46 chromosome in the ovary and the testes. These cells, you'll talk about this a lot in the end of 201 when you talk about, is it 201 or 202? It's 202. Sorry. The end of 202, Bio 202, when you talk about reproduction, the reproductive system and development. But these 2N cells in the ovary and in the testes, respectively, are going to undergo meiosis. And what's going to be produced is a haploid egg or a haploid sperm. At fertilization, those two critters meet up and you get another diploid zygote. And then what we're going to see is this diploid zygote is going to undergo mitosis after mitosis, and we're going to develop a mature organism. Okay. It's this alternation of meiosis and mitosis that allows us to produce the life that we know. There are three main types of sexual life cycles, and they differ on where meiosis happens and where fertilization happens. So I don't need you to know the major ins and outs. As we go through these slides, I'll tell you, hey, I need you to know this. Hey, I need you to know that. Sorry, checking a text, making sure everybody's okay. But here's what we need. So in animals, meiosis produces gametes. Okay, those gametes don't undergo any additional cell division before fertilization. So I have a somatic cell, it produces a gamete. That gamete is just itself, it's a gamete, it's a haploid cell until we have fertilization, and then we get a diploid zygote. Okay? There are two other ways that this happens. Okay, so animals are this guy, then we have plants and some algae, and we have fungi and some of the protists. And I only emphasize this one because this is the one we'll focus on. So I'm going to step back for a moment just to briefly touch on these. So in plants and some of the algae, we have this mature diploid multicellular plant. And on that plant, we have what's called a sporophyte. It's a 2N cell. And what this cell does is it undergoes meiosis, and I get a 1N. Okay, I get a haploid cell. And then this haploid cell keeps doing mitosis. So what ends up happening is this haploid cell just copies itself over and over and over again until we have a multicellular organism. We have a 1N haploid multicellular organism. It's called a gametophyte. It's a plant. It looks like a plant. Okay? And then what ends up happening is we get fertilization from another gamete. Well, it's not even a gamete, it's a sporophyte. We get fertilization from another sporophyte and we form a 2N individual, and then this individual is capable of reproduction. These individuals here are not capable of reproduction. They're not diploid. That's in plants and some algae. In the fungi, they do something even a little bit similar but different. Okay? So they have a zygote. Okay? So a zygote is a fertilized egg or a fertilized 2N. It will undergo meiosis. We'll get a 1N, two cells of 1N, and then that 1N again, like the plants, undergoes mitosis. And so what ends up happening is we form, just like we did over here in the plants and some of the algae, what we end up forming is a haploid unicellular, because we're fungi and some of the protists, a unicellular organism. Okay, sometimes they're multicellular, but it's easier in your brain if you think about them unicellular until you get it down and then add the multicellular in. These guys will undergo mitosis. They'll hook up with another unicellular fertilization, and then we have this fertilized zygote, but the zygote doesn't undergo mitosis at all. So in the plants, that zygote underwent mitosis, right? And in the animals, the zygote undergoes mitosis. But in the fungi and the protists, it doesn't. It just goes straight into meiosis and starts over again. Like I said, I don't need you to know the details of these guys for this course. I need you to know that they exist. I need you to know that plants and some of the algae are different than animals. I need you to know that the fungi and some of the protists are different from the other two. But what I really need you to get a handle on here is the animal version. Okay, so I have this individual, right? I have a zygote. I have a fertilized egg. That zygote undergoes mitosis, 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 mitosis. We get a diploid multicellular organism. It grows into sexual adulthood or sexual maturity. One of the cells, one cell type from there will undergo meiosis and will produce gametes. Okay, one of those gametes gets fertilized with a gamete from the opposite sex. We have fertilization. We get a 2N zygote. Mitosis, 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 mitosis. Okay. Words to help you understand the plant version. So alternation of generations, meaning that the life cycle includes both diploid and haploid in the multicellular stage. A diploid organism is called a sporophyte. A haploid, it makes haploid spores. So the thing that a sporophyte makes is a spore. That spore will undergo mitosis. It'll become a haploid multicellular organism called a gametophyte. That gametophyte then makes gametes by mitosis, and then the fertilization of the gametes results in a diploid sporophyte. It looks like this. It's the thing we just talked about. I just kind of broke it out for you. Here's your fungi cycle. Looks like this. Just need you to know that they're different. Okay, so depending upon the type of life cycle, when you do meiosis is different, right? So it can happen when you're haploid or you're diploid, okay? But here's the thing that's true across the board. Only diploid cells can undergo meiosis, and that should make sense if you're working this through. When you do meiosis, you end up with half the chromosomes that you started with. So if I have a 1N cell, let's pretend it's human for a moment. If I have a cell that has 23 chromosomes and I take that 23 chromosome cell through meiosis, I get 11.5 chromosomes. That doesn't work. Even if it was an even number, even if it was an even number, that wouldn't work. It has to be a diploid. It has to have a larger number. It has to have 2N in order to undergo meiosis. If it didn't, just ignoring the numbers, if it didn't, you wouldn't send on a full set of chromosomes to the next generation, right? So you're assuming that all of those chromosomes are necessary, all of those genes have a specific role or an activity that they do. And so if I only send half of those, I'm going to get an organism that doesn't grow wings or legs or eyeballs or whatever because I didn't pass on that gene. Okay? All right. I'm going to pause right here. I want you to go grab your notes and grab your mitosis. Everything you wrote down for mitosis, or if you printed one of the slides with the image on it, I want you to go grab mitosis and have it handy. If you've printed and you have one of those comparison slides, mitosis and meiosis, it looks, I got to go to the end, it looks like, ay chihuahuas. Oh, it looks like this. If you've got one of those, grab it. Okay? I'm going to back up, I'm going to pause, and then, okay, hopefully you had time to either open your book or grab a page that shows you mitosis, and hopefully you've got one that shows you meiosis too, because this is really visual. And in class, we do it with the wiki sticks or the the paper clips or the pipe cleaners, but I can't really do that well in the setup I have here. So I just kind of have to talk you through. Meiosis, so mitosis was one cell division. Meiosis is going to happen with two cell divisions. So we're going to divide the nuclei twice. Okay, and we're going to call those meiosis 1 and meiosis 2. Okay, the two cell divisions that we're going to see are going to end up with 4 daughter cells. In mitosis, we ended up with 2. We started out with 1 parent cell, we ended up with 2 daughter cells, 2 genetically identical daughter cells. In meiosis, we're going to start out with 1 cell. We're going to end up with 4 daughter cells, and they're not going to be genetically identical to the parent, and they're going to have half of the chromosomes. So not only are the chromosomes that it has not going to be identical to the parent chromosomes, but it's going to have half the number as well. Okay, we're going to go through it kind of step by step, but we'll talk it and then we'll do it. Okay, so the first cell division in meiosis is meiosis 1. This is when the homologous chromosomes separate. So think about it like this. Remember those homologous chromosomes? So I'm just going to draw a really simple cell. I've got to find my blue here because we started with blue before. So I'm going to draw this really simple cell. And I have chromosome 1 from mom, chromosome, and I'm going to do them as numbers just to make it easier. Chromosome 1, 2, and 3 from mom. I'm going to have chromosome 1, 2, and 3 from dad. And it's going to look something like this, right? See how I've put the 1s together, the 2s together, and the 3s together? That first one is not an 11, it's 2 1s, okay? And then it's not 22, it's 2 2s, a mom and a dad 2. Those guys are homologous chromosomes. The 1 from the mom, the 1 from the dad. The 2 from the mom, the 2 from the dad. The 3 from the mom, the 3 from the dad. And in meiosis 1, we're going to separate these. So if this was your cell, okay, those are from your mom and your dad. Okay? All right, let me let that sink in a second. Those are from your mom and your dad. You've got a purple from your mom and a blue from your dad. Okay, so we're going to separate those guys in this meiosis 1. Remember that each one of these is made up of 2 sister chromatids. So I'm going to draw it just a little bit differently to help you with that. And it looks kind of funny because I use numbers, but just kind of bear with me. And they don't really come out being the same exact size, but they're connected at the centromeres. So what I kind of do is I flip them around to be backwards from one another so you can see that, and it makes the numbers look kind of funny. Right? But you're going to get the idea. So remember, there are 2 sister chromatids. Okay, and then in the meiosis 1, I'm going to split them. So these 2 guys are going to go to separate cells. The 1 and the 1 are going to go to separate cells. The 2 and the 2 are going to go to separates, the 3 and the 3s. And then what's going to happen is I'm going to, I should have changed that color. What's going to happen is I'm going to change up, and we're going to separate the sister chromatids. I need to draw this better. Hang on. Give me just a second. Okay, I've got 2 of each. I know that's ugly, you guys. You can laugh. Feel free to laugh. It's okay. I'm laughing on the inside. Right next to the crying on the inside. Okay, so we're going to separate the homologous chromosomes, then we're going to separate the sister chromatids. We're going to do this in class together. Don't freak out. Okay, so at the end of meiosis 2, we're going to have 4 haploid daughter cells. Okay, we'll work it through. All right, so here I have a homologous pair of chromosomes. Each one of these guys, I have 2 chromosomes. I have a homologous pair. Each one is made up of sister chromatids. So in interphase, they duplicate. That's how I get the sister chromatids. Okay? Then we go through meiosis 1. And what's going to happen is they're going to separate. The two sister chromatids that are blue are going to go to one cell, and the two sister chromatids that are red are going to go to a different cell. Okay, so that on the left, I still have two copies of that chromosome, right? I have the two sister chromosomes, and remember they're genetically identical, but they're two sister chromatids, and because they're still connected to each other, we call them a chromosome. This is where it's tough. This is where you just got to hang with me and work it through and write it out. On the right, on the red, I still have two identical copies of a chromosome. I have two sister chromosomes of red, and they're connected at a centromere. Yes? Okay. Now, in meiosis 2, over on the left, those sister chromatids are going to split. And on the right, the sister chromatids are going to split. So that what I end up with are these 4 cells that are different. They're genetically different. Okay? Okay. Meiosis 1 has an interphase in front of it. That's when the sister chromatids replicate. There is no interphase before meiosis 2. Let me say that again. There is no interphase before meiosis 2. Okay? Remember that sister chromatids are genetically identical. Remember they're joined at the centromere. Remember that a single chromosome replicates, forming 2 centrosomes. I'm going to pause the video because we're at 16 minutes. I'm going to come back and we're going to do an overview of this process.